Understanding the mystery of how we develop is beginning a revolution in the way we treat illness and disease. The clues are coming from the very stuff of life.
The major challenge in the 21st century is how we're going to be healthy in our old age. We now stand at the threshold of what might be one of the most exciting periods in medicine.
The aim of all of this research is to be able to offer a treatment to a patient with a disease like, for example, diabetes or Parkinson's disease, heart attack, all caused by damage to cells which are not repaired or replaced.
We might be able to take skin cells from a patient, perhaps just by scraping the inside of the mouth, grow those cells in the laboratory, convert them into nerve cells or heart muscle cells, and reintroduce them into a patient.
Well, we're talking about what is loosely called tissue engineering, and essentially tissue engineering might be used to replace any human tissues which are damaged.
I mean, this is quite astonishing. This is really a transformation of so much of medical practice.
Will we harvest our own cells to restore damaged tissue, or even regenerate whole organs?
Understanding cell development will open up very important new areas of medicine that we can only just begin to imagine.
The dawning of a new millennium could see the rise of a new age in medicine. The seeds of which were sown in an unlikely location, a quiet Scottish village. In 1997, the Roslin Institute near Edinburgh announced the birth of the first cloned animal. Dolly the sheep became an overnight sensation. The British scientists who made her also found themselves in the world's spotlight.
Dolly really is a superstar. She must be one of the best known animals in the world ever. And I don't think that's surprising, really, either for scientists, because for the first time we'd shown that we could take a cell from an adult animal and then make a copy of that adult animal, or to the general public, because of the fact that this is such a new and exciting observation.
She's rather remarkable in that she's very familiar. She's photogenic, at the same time she's a very normal sheep and an extraordinary scientific achievement.
But not all the publicity from cloning Dolly was welcome.
A lot of the coverage initially was full of speculation about human cloning, and there's a tremendous amount of interest.
There has been a continuing media interest in the possibility of using this technology to make copies of people, perhaps a dictator, perhaps an army of identical people. These ideas are really just nonsensical. If you did actually make a copy of a dictator, they might very well turn out to be a nice person, because our personalities are really only half determined by genes and half determined by environment.
An understanding of technology, an understanding of science is crucial to our well-being. And what troubles me so much is that so much of this is actually inflamed by the media, who fail to look at the basic knowledge that we should actually have.
So how do you make a clone like Dolly?
The way that you produce a clone is to take two different cells. You have to have an egg, which you take from a sheep at around the time when normally she would be mated. You remove the genetic information from that egg.
Bill Ritchie, an expert in micromanipulation, using a pipette no wider than a human hair, extracts DNA from the egg. It glows white under the microscope. This leaves just the cytoplasm, or egg white. You then take a cell from whatever you want to clone. For Dolly, a mammary cell was taken from a 6-year-old sheep. The donor cell is starved, which puts it into a quiescent state, similar to hibernation. This allows its genes to be reprogrammed.
It was that particular part of the process that was most unique to the scientists at Roslin. Lots of people had tried to do this, but had failed to actually get the reconstructed egg to develop.
The same small pipette can pick up one of the donor cells, in the case of Dolly, grown from the udder of the donor ewe, and place it next to the egg.
The hibernating donor cell is injected into the stripped-down egg, which amazingly reprograms it to behave like an early embryo cell once again. The reconstructed egg is placed between two fine electrodes and given a mild electric shock.
What an electric current does is it fuses the two cells together, so it brings in the extra genetic information, and it jumpstarts the egg into activity.
The reconstructed egg behaves just like any egg fertilized normally, and the cells divide into an embryo.
It's our role to put these embryos back into a surrogate mother, and in order to do that, we actually take the animal, give it a general anaesthetic again, and pop the eggs back in. At the scanning stage, we knew she was special in some way, but not the significance of it, not that she'd been produced from an adult cell.
By reversing the biological clock of an adult cell, the Rosalind scientists rewound its cycle, taking a specialized cell back round to the start. The team are now planning a medical revolution.
We all start off as a single cell. By the time we're born, there are perhaps several billion cells in our body of perhaps 200 or 300 different cell types. Heart cells, muscle cells, nerve cells. Every cell that has a nucleus, and almost all the cells in our body do, will have a full genetic code, have a full complement of 100,000 genes. But most of those genes are switched off. A muscle cell, for example, doesn't need the genes that, for example, code for brain neurotransmitters. So in most of our cells, most of the genes, perhaps 90% of the genes, are switched off.
That's what enables the embryo as it develops to produce muscle and brain and intestine and all of the different organs that are necessary for a person or an animal. And the view at the time when we began the project was that it was not possible to reverse those changes.
So, irrespective of how long a cell has been specialized, however long genes might have been switched off, it seems possible to reactivate them and turn a cell's biological clock back to zero.
The most important element of the cloning experiment was the notion, undoubtedly, that you could program the nucleus by the technique of transferring it into a new cell, i.e. an egg. And that, I think, is fundamentally important.
If adult human cells can be rewound back to the beginning, instead of making a whole organism like Dolly, they could be made into the tissue needed to repair patients' bodies after accidents or to cure disease. When the biological clock is first wound, the single cell divides and divides. Inside this early embryo are cells with a unique ability. Human stem cells have an amazing power to make all the tissue in the body. Scientists in Roslin are now focusing on how to control stem cells to grow any tissue at will.
Tissue engineering might be used to replace any human tissues which are damaged, though it'll be much easier to replace tissues which don't have, if you like, a structure of an organ. So it may be very difficult to replace a kidney or a heart or a lung using tissue engineering, at least initially. It might be relatively easier to replace liver tissue or possibly fat tissue or maybe even muscle tissue, because there you might expect, given the right genetic influences, to get the tissue to grow in a uniform manner, mimicking how it would do during normal human development.
Many of us will suffer from diseases like diabetes, leukemia, stroke, or Parkinson's. If any one of our cells starts to fail, this forms the basis of degenerative disease.
I had a tremor in my small finger, my little finger, and I thought I had a sports injury. So I went to the doctor, and after I actually told him that my father also had Parkinson's and I hated tremors, he clicked and said, sit down and we better send you to a neurologist. I had the diagnosis as young onset Parkinson's, which was a hell of a shock.
But could Rosalind's dream of replacing skin, brain or blood cells in diseased bodies ever really work? In Cambridge, scientists think it will. At the internationally renowned Brain Repair Centre, they are already developing new nerve implants to help patients.
Traditional approaches to treating neurodegenerative disorders have relied largely on administering drugs produced in order to replace chemicals missing in the brain. A good example of this is Parkinson's disease, where the chemical missing is dopamine, and the loss of dopamine causes a rigidity and problems with movement. And the analogy is rather like losing the oil in the engine of a car. And once the oil's been lost, the car seizes up and it can't move. If you put the oil back in, the car will drive off again.
At the moment, drugs are used to target damaged cells. But this means soaking the whole brain in the missing chemical when only one part needs it. Cell therapy is a new approach. In the future, tissue will be grown to match the failing cells and then transplanted into patients.
In cell therapy, we actually replace the dopamine cells that are lost rather than giving the chemical back.
The current work in Cambridge is with rat models to find a cure for Parkinson's.
And the recent excitement with stem cell biology has meant that this may be a step closer. Here in Cambridge, we work with cells that we isolate from the fetus, and we grow these cells in the culture flasks as these sort of spheres, and they get bigger and bigger. And each sphere may have up to 100,000 individual stem cells within it. And so we take a whole sphere and we put it into another culture dish, and we ask it to stick and start producing brain material.
Here, the stem cells are growing into brain cells.
It's almost poetic when you see cells. They look like trees growing in the garden. They've got branches everywhere.
The cultured stem cells are then simply injected into the area of the rat's brain damaged by disease. Miraculously, they grow into mature neurons in the rats. One fetus can provide thousands of stem cells when grown in culture, but as yet, when transplanted, they fail to produce the magic dopamine. So to treat people in this way is more difficult. For the brain cells to produce the missing chemical, the stem cells must be transplanted from aborted fetuses. But trial operations have given remarkable results.
Neuronal transplant for Parkinson's disease has been going on using fetal tissue for some time, and it certainly does work. It works up to a limited extent. I have one friend, actually, who's an extremely well-known author who developed very, very severe Parkinson's disease, whose life, has been pretty well saved by neuronal transplantation.
Every patient requires 5 fetuses in order to achieve a transplant on one side of his brain or her brain. And so this has really restricted the method and this type of approach to treating this disease to very small centres and very small numbers of patients.
There are quite legitimate ethical reasons and objections to using fetal tissue, because there I think you are tampering and destroying human life.
Alternatives to using fetal tissue for cell replacement therapy recently began at the University of Pittsburgh Medical Center. This is the world's first cell replacement operation for a paralyzed stroke patient. Instead of fetal tissue, surgeons are experimenting by injecting cultured neural cells into the brain. A needle syringe directs the cells in and around the area of the stroke, using the same technique as the Parkinson's trials. Research is at an early stage and relies on growing a large batch of the same cells to treat everyone. But could treatment be personalized to grow tissue from the patient's own healthy cells? The Rosalind team think so. The latest research in Scotland hopes to tailor the treatment individually. Using a technique similar to the cloning of Dolly, they want to fuse a patient's healthy cell with a human egg. After a few days, the stem cells could be extracted for transplantation. As a clone of the patient, the new tissue would be a perfect match.
The vision would be that we could take a healthy cell, a skin cell, for instance, from a patient who's starting to suffer from Parkinson's. We would then use techniques that build on what we know from animal cloning to reprogram that healthy skin cell into a new population of replacement cells for the damaged cells in that patient's brain.
The work that the Roslin are doing at the moment is fascinating and very interesting for us, because we'd like to actually reprogram our human stem cells in order to make them into dopamine neurons.
Even without ethical concerns, the difficulty of getting lots of human eggs to reprogram our adult cells is not a practical way to treat the millions of patients who could benefit. The miracle of Dolly could once again hold the key. Roslin Institute think that instead of eggs, stem cells could reprogram the patient's cells.
If we can identify what it is that an egg does to the genetic information, if we can find ways of mimicking it without using an egg, then we will have ways of being able to offer people, for example, the nerve cells which are needed to correct Parkinson's disease.
Once a human cell had begun its life cycle, the final destination always seemed permanent. It was destined to stay a blood, muscle, bone, or skin cell. But Roslin's research shows cells can be transported back to the beginning and reprogrammed to go in a new direction as a completely different new cell.
Cell therapy will depend on being able to take a healthy cell from the patient and reversing this process right back to the beginning before we then control its development down the particular path that we want, so that we produce islet cells for patients with diabetes, neurons for Parkinson's, heart muscle for people who've had heart attacks. But it all depends, first of all, on taking the genetic information from a healthy cell and making it go back to the beginning before it comes up along the route that we want.
It seemed as if the only way to do it was to use embryo tissue, because only embryo tissue has the potential to grow into any other kind of tissue. Well, now with the techniques that produce Dolly, in theory it becomes possible to turn any kind of tissue into any other kind of tissue. So you miss out the embryo stage altogether. So it really doesn't seem to me, if you use this advanced technique, that you raise any real ethical problems at all.
It may be possible to identify people who are at risk from the sort of degenerative diseases that cell therapy could treat. And in those circumstances, you could imagine for a proportion of the population, banking cells. We could take cells, for example, from the umbilical cord of a newborn child and bank those cells in the expectation that they might well be needed perhaps 30 or 40 or 50 years later.
In the future, reprogramming our own cells will make growing fresh supplies of simple tissue possible. Cell replacement therapy could finally provide truly effective medicine. But would it really be possible to engineer whole organs or large body parts?
There are better scientists than myself who seriously believe that growing organs in culture may be possible. Whether we will grow a heart or a lung or a kidney in my lifetime, I think is fairly dubious. But there are serious attempts being set up now in laboratories to try and do that.
It's a tremendous thing to be able to produce cells of any kind you want in culture from any other kind of cell. That's amazing. To produce organs, whole organs, is one or two or several steps further on. How are you going to turn this sort of sheet of cells into a three-dimensional organ? One way that's been proposed is to build a kind of plastic model of the organ and then let the cells grow over it. But the plastic model would be biodegradable, so soon that would disappear and all we'd be left with would be this facsimile of the original organ.
Let's imagine a heart, for example. So you need muscle, you need nerve, you need various bits of connective tissue, and you need the cells that line the blood vessels at the very least. So you have to get different cell types. You then have to get them to arrange themselves in a three-dimensional structure, which is immensely complex, and grow out to something which is larger by far than anything which would ever been grown at the present time. So it may be possible one day, but I honestly think that is very, very far into the future.
And a lot, I think, will depend on our understanding of the genetic control of how these particular organs develop. That may be the key to much of this work.
I try never to say never about what may happen because of scientific research, because after all, remember that people said just a few years ago that it would be impossible to produce a clone like Dolly. But having said that, I do think that the idea of trying to grow a whole organ is really very ambitious.
We all start life as a single fertilized egg, which grows to produce a whole person. Stem cell research is at the core of understanding that process. Just a few years ago, the idea of taking patient cells and turning them into any tissue needed would have seemed pure science fiction.
This area of technology has the potential for abuse, but there are much more everyday technologies which can be abused. The chlorine that cleans up our drinking water was used as gas in the First World War battlefields, as a weapon. There are many bacteria that we use to make antibiotics that could be used as weapons. So I don't think there's anything unique about these new technologies in terms of their potential for abuse.
Cloning does raise a large number of ethical issues, and some people have suggested that what we do is unnatural or is playing with God. I think mankind has been trying to improve on nature for 5,000 or 10,000 years, and certainly in the last century we've had some major medical breakthroughs that have benefited everyone. Vaccination, antibiotics, for example. And we believe that our work is very much in that tradition.
People very rapidly accepted transplantation, and they very rapidly accepted in vitro fertilization. It was only within a few years of Louise Brown's birth that people widely believed that IVF was a good thing.
In the very long term, these technologies are actually going to transform all of medicine. And when we're talking at the moment about replacing damaged tissues, replacing damaged organs, in a sense that's only the beginning. The real point is that as all the technologies come together, we're coming to understand the very fundamental processes, why cells grow the way they do, why they differentiate, why cancer cells arise. That's one of the most intractable things of all these days. Why, for example, things like AIDS just take off. Well, when we really understand how the cell works, we will be able to get on top of all these diseases. And this kind of technology is taking us into the areas of knowledge that will enable us to do that.
I come from a tradition which believes that that part of that divine spark is our intelligence and our inventiveness, and that was actually a God-given tool, and that to fail to use that actually is a mistake. It's true that the knowledge can be put to evil use as well as to good use, but if you don't have the knowledge, you can't decide. And the key aspect of our morality is our ability to have free will, the ability to choose between good and evil. So without that knowledge, you can't begin to choose. So you're not human.
Our choice has brought us the power for greater biological understanding of exactly what makes us human. This new knowledge from the research in Cambridge and Roslin could give us the ability to alter the fate of future generations.
These expectations come to fruition, then it'll be a tremendous legacy for the future from essentially a Scottish sheep.